without any further ado, let's welcome Professor Ju. And I know all of you have been waiting to hear from him. I would introduce Dr. Ji-Hyeon Ju, who is a professor of rheumatology at the Catholic University of Korea. He earned his MD from the College of Medicine at the Catholic University in 1997, followed by an MS in 2008 and a PhD in 2012 from the same institution. Dr. Ju currently serves as the director of the academic committee of the Korean Society for Stem Cell Research. And in addition to his academic role, he is also the CEO of YiPSCELL, a biotechnology community focused on developing stem cell based therapies. The company specializes in generating stem cell lines from both healthy individuals and patients using advanced induced pluripotent stem cells, iPSC technology. Dr. Ju also contributes to national regulatory and scientific efforts as a member of the Advanced Biopharmaceuticals Specialist Committee at the Korea Drug Safety Management Institute, as chairman of the Review Board for Pharmaceuticals and Medical Devices at the Ministry of Health and Welfare, and as a biomedical expert member of the National Science and Technology Advisory Council. So it's our immense pleasure to host Dr. Ju today, and we are excited to learn about designing health in space, the future of iPSC-based space medicine. So give a big round of applause for Dr. Ju, and we are really, really excited to host you today. So I would stop my screen share and over to you, Dr. Ju.
Thank you for your kind and long introduction. And I have some positions and directions in our institute. And I prepared my presentation in a mixed form of English and Korean. So please understanding my situation. So I am Korean, so I'm not good at speaking Korean in English. So please understand that. Yes, let me share my slide first.
Sure. We are really happy to host you, and please feel free in your communication. For any international participants who are in the meeting today, you can use the chat section to put your questions in English, and we will take them up after Dr. Ju's presentation is done. So thank you so much once again, and over to you, Dr. Ju.
Yes, let me introduce again. My name is Ji-Hyeon Ju, and professor of Seoul Saint Mary's Hospital, and also professor of the Medical College, and also CEO of YiPSCELL, stem cell company. My topic is about designing health in space and future of iPSC based medicine. My content is like this in backgrounds. Why need artificial blood product? And followed by problems. We facing some hurdles in artificial project. And third is what is the solutions and overcoming through novel solutions. And finally, perspective, the clinical and industrial application of space biology. And first, backgrounds. I'm so sorry that this is Korean slides. However, the transfusion history is long history. However, it started at Korean War, 1950. And maybe you know that war needs a lot of transfusion science. And since that Korean War, a lot of the events are followed in like the National Blood Institute, and the nowadays a lot of transfusions are performed for the patient in South Korea. However, we having some big problems because of you know that Korean is most very notorious for most fastly aging society. It means that transfusion it becomes more difficult to get the blood from young adult because aging population could not provide blood for transfusion. So Korea is facing that shortage of the transfusion amount. Maybe you know that in Korea is have a big problem of rapidly aging and yeah. So and another big events you know that COVID-19. The COVID-19 period it was almost impossible to get transfusion from the population. So Korean government started to think that transfusion maybe could be uh, yes, yeah, the lack of and the shortage of transfusion could be overcome by some advanced science. Maybe you know that artificial blood. And also, we need more and more blood for various kinds of blood disease. So yes, lots of the blood mm, blood replacement could be done by some kind of artificial blood, some kind of this. I'm sorry for this Korean language, and the scientists have developed artificial substitute, such as some kind of bead and some kind of chemicals, but they have problems of safety and efficacy. So in clinics, there is no way except transfusion science. But you know that for the large war, some kind of the some emergency, government prepared that some chemical-based the blood substitute for some kind of special event. However, in normal condition, Korean society, yes, depends on the transfusion nowadays. This is background of the international and domestic artificial blood science developing history. Maybe you know that uh, blood cell could be generated from various cells, stem cells, from the cord blood or some kind of embryonic stem cell, iPS cell, and some, yes, it's the science have could, yes, they made it possible to generate a red blood cell from stem cell. However, it also have some problems because the final stage of the RBC generation, it is essential to enucleate from erythroblast. However, it is very difficult to extract nucleus from erythroblast. It is very technical barrier. And another big problem is efficiency of erythropoiesis. This is very difficult to, it's very expensive, expensive to make it RBC from iPSCELL. It is very expensive. So scientists try to develop economic way of erythropoiesis from stem cell. This is comparison table. This one is embryonic stem cell. This is induced pluripotent stem cell and cord blood and peripheral blood and immortalized cell line. I choose, my specialty is induced pluripotent stem cell. It has the merits of the safety issue and some kind of the scalability, but maybe you know that iPSCELL has the some very proliferative and it means that some kind of it means that some tumorogenicity, cancer development. So we have to get the evidence of not no tumorogenicity. It's a very important issue in iPS cells. And some problems. And then it is my project before space medicine. Uh, I'm so sorry, in Korean language. And clinical grade iPSCELL drive artificial blood commercialized industrial plan. And for 3 years, we have to develop that. We have to establish clinical grade source iPSCELL lines. And I have to make it more specific and industrialized standard of protocol for the artificial blood cell. And we get the RH minus, RH negative, O type is universal donor, and we could generate iPSCELL lines. It means that cell banking is possible, and we make it. We can make just one bottle of iPSCELL. We can generate the whole blood for all the all over the world. It can expand it to worldwide widely. And so it's very effective way to some commercialization and industrialization. And cell banking is needed for industrial way and proliferation culture and differentiation culture and inoculation and scale up and commercialization process. We have to develop these processes in details. After, after establishing this process, we have to clear these steps. It's for the IND. It means that investigational new drug process for the Korean FDA permission. So for the permission from Korean FDA, we have to prepare some documents and some data for reviewing from regulatory bodies. It is very difficult time. Maybe as a scientist and as a clinician, uh, I have very difficult time with the Korean FDA reviewers for 5 years. And finally, we get the IND permission from Korean FDA. I started my own clinical, yeah, it's not artificial blood. It's for the injectable steroid for osteoarthritis treatment. And last month I started, but it is very difficult way. I don't recommend to follow my way. We have to fight against the regulatory bodies. This is very difficult. Anyway, and this is overview. We have to prepare the raw material and we have to manufacture it and we can yes we can make it the final product. And for 5 years, it's a Korean way. However, we prepare some cell banking and we have to set up the culture the way and the way of the inoculation technique uh, and the scale up the scale up and the finally we have to prove that the quality control the standard we have to establish. And yes, maybe in Korea, we don't, very small population of the Rh negative or blood type O per population. So we, with the help of the Korean government, we selected four donors of the universal blood type, and we could establish. We can, yes, maybe you may not know. iPS induced pluripotent stem cell could be generated from blood of the some population. So yes, we could retrieve blood from four persons and finally yeah we were successful in establishing iPSCELL banking in GMP facility. Have you heard about GMP facility? A good manufacturing product means that the germ-free and very strict environmental limitation is like some space and it is very essential facility for biological drug production, and we have to generate these cell lines in that facility. And this is the manufacturing process. iPSCELL and could be generated into embryo body, and maybe you know that yolk sac like structure could be primitive as voices happens in this organs. We can make it many yolk sac like organoids and induced hematopoietic stem cell could be generated, and finally, ILBC could be generated. This is processes, and iPSC, EB, and mini yolk-sac, and IHSC, and ILBC. And you could see that very small amount of blood here uh, in petri dish and tube. We can generate this kind of artificial blood from stem cell. very small amount, very expensive way. You could see here, this is the yolk cell-like organoids, iPSC drive yolk cell-like, and yolk cell produce lots of hematopoietic stem cell like this. So we mimic that human body erythropoiesis system. And the another way to promote erythropoiesis process, it is bioreactor. Not manual way, but bioreactor is one of very effective way to scale up. It means that cost-effective way. And in Korean, KBS, Korea Broadcasting Station, and it's a national as National Broadcasting Center, they yes, they get the our achievement in very big way of the news, and they say that bioreactor is the future way of making the artificial blood in South Korea. This is the one IBC pack. 2 years ago, we make it one pack with three $3 million point $3 million, and last year and one and a half, yes, and using the bioreactor is below $1 million. But maybe you know that for one pack of RBC, it is even the richest country could not afford. This is a very impossible way to provide blood for the patient, very expensive. So we have to find a solution in overcoming through novel solutions. It means that the iPSCELL nowadays from the Mayo Clinic or some NASA, they have developed way to culture stem cell in space, in ISS, in space microgravity, because of very recent years, 2024, they have lots of experiments on the stem cell culture in space, in ISS. And NASA, they have several programs about that. And even in some Mayo Clinic or some Cedar Sinai, maybe you know that Los Angeles, and they have Los Angeles are focusing on iPS culture, and Mayo Clinic they are focusing on the hematopoietic stem cell culture in ISS. So they are interested in the culturing this stem cell in space. It means that the in space have some merits because of the maybe you know that iPSCELL EB, the yolk cell-like structure, hematopoietic stem cell, and we use that not 2D culture, but 3D culture is more effective way. And not static culture, but suspension culture like bioreactor is more effective way. So, however, this is the most recent advanced way of culturing stem cell for erythropoiesis. However, very low efficacy, and it means that a very expensive way to generate RBC. So in Korea, Korean government initiative raised big fund for the Alpha H, and it means that mm, noble way is totally noble way to overcome for very big hurdle in science world. So stem cell on Earth like this. They due to gravity, stem cell if we culture in 3D way or 2D way, they facing the face against each other because of maybe your body is composed of in not 2D way in 3D way. So cell interact each other like this. However, if we culture in petri dish, the cell should be stuck to the bottom and very artificial um, environment. However, we if we reduce uh, not Earth gravity into low gravity like this, and if we make it microgravity, and we can make it cell happy. So with yeah, like this way. So in this field, scientists have developed lots of facilities and device like this. Random positioning machine is artificial simulated microgravity. We make it random positioning machine for if we culture cells in this machine, they'll experience microgravity. And also, this is another type of rotating wall vessel. It is also simulated microgravity machine, and we yes, we have these machines in the lab, and we could culture cells in this machine. We could yeah simulate microgravity in cell culture. Maybe it is the best way to get the real microgravity, and we have to we have to get cells into the space. It is most effective way. However, maybe it is also another very expensive way to get some experiments. So nowadays, many scientists try to simulate microgravity with this kind of machines in the lab. And in Korean initiative, we have uh, uh, yes, we with a collaboration with the space launch satellite company, and we put the bio cabinet into satellite, and we would like to prove our postulation of the microgravity could be uh, very good environment for erythropoiesis in space. And with a collaboration with space satellite company Lumid, we have worked together for 2 years. This is the overview. We generate iPSC line and the make EV and hematopoietic stem cell generation and erythroblast and erythrocyte. It is overview, and we will simulate microgravity in these processes. And from the iPSC, we can make it yes, using the random positioning machine and yes, it's clinostat. Clinostat is the name of the micrograph simulation machine, clinostat. Using clinostat, we proved that iPSC could be more proliferative, and also this kind of hematopoietic stem cell could be well nurtured in space, and also maybe at the beginning of my talk, I talk about the very big hurdle of inoculation hurdle. The nucleus extraction is a very difficult process in artificial culture system. So using microgravity, we will simulate these inoculation processes in space. So we nowadays we have developed the bio cabinet. It's kind of bio module for the iPSC culture and hematopoiesis culture, and three kinds of biomodule we have developed. So make it together three stack machine, one cabinet, and we put into stuck into the satellite, and we will launch it in 2027. This is a space bio cabinet, and another name is space bio module. And it is all about the cell culture and this waste tank and culture media, culture nutrient, and stored here, and the oxygen and nitrogen here, and we put together into this test space. Cell culture here we put the some kind of the oxygen and nitrogen and carbon dioxide and new culture nutrient here. And of course, this is a non-human flight mission, so we have to equip and with some kind of monitoring devices here, some kind of pH monitoring and vision unit. This is needed for that, and waste tank is needed for yes, get rid of this waste in this culture system. And this is collaborative work in Seoul Saint Mary's Hospital and IPSCELL and Lumit, and work together for this project for 3 years. Nowadays, we generate new protocol for space culture, and because in earth ground culture, for the better adapted space culture, we have to change the nutrient and some kind of culture condition. So nowadays, we are focusing on the new culture system for space, and also we have to culture's period is shorter for culture flight missions. So usually it is needed for 1.5 months for complete erythropoiesis. However, we would, yeah, our first the purpose is to shorter to the maybe 2 or 3 weeks for the flight mission. And another way is the kind of the uh, bioreactor, because in earth ground, yes, men can work by manually. However, in space, it is difficult to, because space astronaut sometimes are not good at experiments. So fully automated bioreactor is better for space medicine. So nowadays our processes is make it better for some this kind of the automated bioreactor programs. And maybe you can see that it is vision unit for colony forming unit assay, and with the help of the company IPSCELL, the microgravities simulated machine clinostat, we have equipped 5 machines here. This is commercialized product, Clinostat 2, and also we yes, developed by ourselves three Clino Y, and this is we're using 3D bioprinting. And so we developed three by ourselves and two we bought. Nowadays, we are focusing on the initial the conditions for culture condition in clinostat. This kind of clino Y and clinostat. Clino Y is a random positioning machine, and clinostat 2 is a vertical wheel type of the clinostat. So this is iPS culture. You can see that using this clino Y, you could see that at OCT4 and SOX2 and lane 20, especially OCT4, you could see that this is pluripotent marker. So this is pluripotent marker means that stem cell is more effective in simulated microgravity situation conditions. And also this is vertical wheel type. If we culture this embryoid body, you could see that we could make it EB in this machine. This is very preliminary data for condition setting. So but you could see that this is the yolk like material is very bigger. I have never seen this kind of biggest yolk organoids and in the clinostat machine. We could not see in 2D cell culture in earth ground. However, using microgravity simulation, we could generate a biggest yolk structure in this machine. And another very interesting observation is a brain organoid. And if we culture brain organoids in this area, you could see that this is the normal gravity and simulated microgravity. Morphologies are different. And finally, this is last week uh, experiment, and students provided me these photos today. You could see that normal gravity, brain organoids very small. However, microgravity, the brain developed very rapidly, more bigger brain we could see in even in the same culture period. And then with the help of the satellite company Lumir, they are developing this designing model for the payload, the satellite. They make some maybe you know that develop model and engineering model, and they have the a lot of some test and certificate for space medicine, and it is some another way, another field we cannot get into because it is very specialized area for making some space specific culture device, and they are very good at this field. So we they helped me to develop this space cabinet for culture for future launching. And it is summary. Random positioning machine, yeah, we have developed by ourselves for using 3D printing machine and the rotating wall, yeah, vessel. And yeah, we bought it from this commercial company from Europe. And how could follow the real microgravity? It is only way is that to like this. In 2028, we will launch not human, non-human flight mission. We will, and 2028, yeah, at that time, we will. Yeah, send our samples to ISS. So in astronaut uh, will do experiment from the iPSC to erythrocyte. In 2028, we have the already contract with NASA. No, not SpaceX, not NASA, SpaceX. So this is the risk uh, factors because of the space radiation and cell DNA alteration. And maybe you know that after launch, we have to 3 or more weeks for the to get into the ISS, and 3 weeks we have to have freeze because for 3 weeks after just after launch, we have to make it cell freeze because a long time is needed to get to ISS, and some kind of the temperature change and the sample retrieval difficulties, and a lot of risk factors are there. We have to make yes make a solution for that, even if it's successful to make it bio cabinet. And this is the I am scientist and I am biologist, so iPSC-cell, hematopoietic stem cell, and erythrocyte are now focusing on. However, this is erythropoiesis and lymphopoiesis. You know that this kind of lymphopoiesis is for very good future application for the maybe you know that nowadays cell therapy for cancer treatment. So CAR T cell therapy, have you heard about that? And CAR T cells could be generated in space because of a more effective way to generate the T cells, and also the viral transduction is very good way to yes, it's very effective in space. Also, protein crystallization is effective in space. It's well known that maybe I think this process could be applicable to space medicine. Yes. Uh, yes, this is the, I am very sorry for this Korean language. And I started artificial blood Korean initiative since 2020 November. Uh, it this cost is $5 million for 4.5 years, and since 2024 December, we get the we have the a very big Korean initiative for Alpha H, the space application of artificial blood in space medicine, and we yes. So my group is system is academia and it's Catholic University of Korea. My research members and my PhD students, and YiPSCELL is my company colleague, and maybe about 60 members. We work together for uh, developing novel way to erythropoiesis in space medicine. Thank you for your attention. Thank you.